All right, so welcome back, everyone. I hope you had a nice tea break. So, we will start with the antibiotic resistance part 2. What we are covering here is we are now going to talk. We learned in the previous one how, what is the mode of action of some antibiotics, what do they do, the wonderful things that, you know, these wonder drugs, as they are called, or silver bullets, as they are framed, how they help in saving lives. Yesterday, you saw how antibiotics increase life expectancy, right? By how many years? 23 years, yes. So today now we are learning about, how resistance develops, how it can spread, ways in which it can be tested, what we can do about it. Okay, so mechanisms of resistance. So bacteria can be intrinsically resistant to antimicrobials due to absence of susceptible target. Okay, so there is no target over there for the antimicrobials in that particular organism. You know what is a target, right? Target is needed for it to, for the antibiotic to act on that target so that it can kill the bacterium, for example. So an example of that is vancomycin um, for gram positives. So vancomycin cannot access, for example, the outer membrane, right? So vancomycin is a big drug. It is a very big antibiotic. So it can't access the outer membrane. So it can't be used for gram-negative bacteria. Okay, so that is why it is used only for gram-positives. On the contrary, colistin, targets the outer membrane. Where you will find outer membrane? Gram-negatives. So it is the one that is targeted towards gram negatives. So bacterial, can you close the door? Is it closed or sorry? Thank you. So bacteria can also acquire or develop resistance mechanisms, and I think you know what I mean by acquire, right? You are acquiring it from outside, or you're developing, meaning maybe you have a mutation or something. So that they can do by, for example, minimizing the intracellular concentration of the drug, okay, by poor penetration or efflux. So understand the sentence again. Minimize the intracellular concentration of the drug. So the drug is inside, so that concentration that is there is very low. If the concentration is very low, then how bacteria will be killed? It can't be killed, right? So that can be by poor penetration. So it will not penetrate, very poorly it can go inside. Or efflux. What is efflux? You know what is efflux, right? Take the antibiotic and throw it out of the cell. Efflux, lots of efflux pumps are there. And gram negatives are very famous for efflux pumps. Okay, most efflux pumps and most efflux mechanisms, and in general, mostly, gram negatives have a very wide range of mechanisms compared to positives. That is one. Second one is modification of target by genetic mutation in the gene or something, or post-translational mutation of the targets. The post-translation mutation is occurring in the target that is happening. And then the last one is hydrolysis of the target. Um, so the target is being hydrolyzed. So first one is mechanism of resistance. So prevention of access to target. Okay. So you're preventing the access to target. So gram negatives are intrinsically less permeable than gram positives. Okay. As the outer membrane acts as a permeability barrier. Everyone knows what is a permeability, right? To penetrate. So hydrophobic antibiotics, example beta-lactams, cross the outer membrane by diffusing through porins. Okay, so porins are channels. These are protein channels by which the antibiotics can cross the outer membrane. So that is one. So bacteria can reduce permeability by loss of porin. Okay, loss of porin, then altering them. So that is partial, it is partial loss. And then third is decreasing porin expression. So that is moderate loss. And that you can see very nicely in that picture on your right hand side. On the right hand side, you can see that you have the drug that is there, blue in color, and you have got the outer membrane, OM, you have got the periplasmic space, PP, and you have got the inner membrane that is there. And what is happening is in the first one, in the first thing over there where it says mutant porin loss, you can see that the porin channel that is there, that is being lost. So, matlab, porin that is there, it's like a door. Okay, imagine a door that is there. If the door is removed and if the thing is sealed, you don't have any entrance for that, antibiotic to come in, no, inside the door. So it is gone. There is resistance that is, okay. In the earlier days, the kings and all, they being build big fortresses, right? For what? That was for prevention of the other people, armies from other, is the same mechanism that is showing over here. So that is first one. Then second one is altering them. So when they are altered, when the door is, twisted or something, then you can see that, okay, partially people can come in or partially the antibiotic can come inside, but it is not at that concentration where it can kill. Okay. And then the last one that is there is, decreasing the porin expression. So the expression of the porin itself, instead of, I don't know, previously maybe it had, I don't know, 10 doors. So instead of 10 doors, maybe it will only have like one door, something like that. So the porin expression is decreased, so moderate. So resistance to carbapenems, resistance to cephalosporins can be conferred by this mechanism. And a very good example, a very classic example of this is a CAR-O protein. Okay, CAR-O protein, you can write it down if you want. CAR-O protein is a protein in Acinetobacter baumannii, which I think you guys know is one of the bacteriums I study the most. And the loss of that protein can lead to carbapenem resistance. So you don't need like enzyme to hydrolyze and do other things. Various mechanisms are there, efflux, and so this is prevention of access to target. Okay. So next one is mechanism of resistance by hydrolysis. Everyone knows what is hydrolysis, right? So enzyme catalyzed modification of antimicrobial is major mechanism of resistance. So first discovery in 1940 was of the penicillinase, which is the enzyme. Don't need to tell you what the penicillinase is going to do. It's going to hydrolyze penicillin. But you have also got cephalosporinases. You have got the last CTXM15, CTXM group of enzymes, or Acinetobacter-derived cephalosporinases, for example, or MC genes that are there. They are all cephalosporinases in different bacteria. Then carbapenem, you have got carbapenemases, NDM, you have got VIM, you have got SIM, GIM. So many different enzymes are there that can act as carbapenemases. So that is one mechanism, that is hydrolysis, where the drug is being broken down by that enzyme. And then the next one is steric hindrance. Okay. So steric hindrance is the modification of the, of that particular antibiotic that is there. So that antibiotic is modified in certain ways. So when that antibiotic is modified, then it can't, affect that particular target. If it's not going to affect the target, then how it is going to kill? It can't kill. And then there are examples like acetyl transferases, phosphotransferases, nucleotide transferases. These are all aminoglycoside modifying enzymes, these are called. Okay, so these are classic examples. So mutations causing resistance now, right? There are mutations that can cause resistance. I gave you example of the gyre-Parsi gene previously, what they were supposed to do. You know, gyre is doing the unwinding, and then you have got the Parsi, which is, removing the interlink between the DNA. Now, what is going to happen? When people go to party, right? I will give you example now. See how I will explain. Don't be surprised. You'll understand what I'm trying to say. You go to the party, right? You're going to party, then sometimes people wear mask, right? When you're wearing the mask, you can't identify who is who over there. So what is happening over here is that same phenomenon occurs over here. So when the mutations are occurring in the gyre-Parsi gene, the uh, the fluoroquinone that is there, it was recognizing that, oh, this was a gyre, this was the Parsi. It can't recognize. So it's almost like they are wearing mask over there. So they can't recognize. So when they can't recognize, then they can't act. And if they can't act, then the resistance is created. Understood, no, what I'm trying to say? So it is very simple, everyone is thinking like, oh, these concepts are so difficult and you are burdened with everything, but I think you understand what I'm trying to teach you. So that is the thing that happens. So these regions that are there, gyre, Parsi, these are called as quinolone resistance determining regions. And these regions and the previous one, the examples that are given, these are very specific. They are very, very to the point. They are not like um, just good to have. No, these are the most important mechanisms. If something is happening over there, then is already established that there will be a problem, okay, or there will be a resistance occurring. So mutations in the small region that reduce the affinity of fluoroquinolones to gyre and Parsi, and then as a result of that, you already know there will be resistance that will be created. So we will also see how easy it is for the bacterium to develop resistance.
Body is basically a petri dish. It's supposed 2 feet by 4 feet. And it's just set is moving bands. At the base of each of these bands, we'll put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's an antibiotic. You stem from that. There's thing that the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And of course, the clip of it, or some thin agar, that bacteria can move around in. Sometimes good because there's ink in it. The picture will appear as white. You see this in the area where there's no antibiotic, up and up that can no longer survive. It appears on the shape. It's resistant to the antibiotic, it's splits, just to compete with other mutants around it. Well, usually the check that it's ready. There are two have to pause and develop new mutations to make it 10 times as much antibiotic. And then you see the mutants repeat this at 100. And alone in days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see how this process of accumulating successive mutations, uh, bacteria which are normally sensitive to an antibiotic, can have enough resistance to extremely high concentrations in a short period of time. That's
So hopefully you got a very good flavor of how resistance evolves, how easy it is for bacteria to develop resistance, right? So determining antimicrobial susceptibility. Everyone knows what is antimicrobial susceptibility. It is a test to determine whether the bacterium is, you know, sensitive, resistance, or it has intermediate level of resistance to an antibiotic. So gold standard for characterizing the susceptibility of organism is by determining the minimum inhibitory concentration, MIC. You might have heard about this uh, phrase before, or uh, these words, which is MIC, minimum inhibitory concentration. So MIC of an organism is the lowest amount of antimicrobial, okay, lowest amount of antimicrobial required to stop visible growth, okay, required to stop visible growth. So there is no growth over there. Remember that it is MIC, okay, minimum inhibitory concentration. There is also MBC. I have not highlighted over here, but there is also MBC, that is minimum bactericidal concentration to kill, you know, completely. So we can find about MIC in different ways. So this method is, so this is not for MIC, but this is for the susceptibility testing. And this is a cubivoid diffusion, disk diffusion method. Everyone knows what is a disk diffusion, where you have your plate, agar plate, which is the medium for bacteria to grow, and you are applying um, a concentration of bacteria at certain McFarland standards. You're spreading it on the plate, and you're putting what is known as different discs of antibiotics. And what you see is, you can see the zone of inhibition, right? You will see that the disc, from the disc, the antibiotic will diffuse out, and it will start diffusing out, and then it will kill the bacteria at certain concentration, okay, at certain diameter. So the zone of inhibition is always measured in millimeters. So that is the entire diameter that you see in the circle in the inhibitory zone. This is always measured in millimeter, not in centimeter, not in any other thing. It's a very standard practice, and that can be interpreted using the UCAS guidelines. CLSI guidelines are there for certain bacteria, and you can classify the organism as sensitive, resistant, or intermediate. Okay, so this is by far the most useful method across the world. It is very simple, and it is also very cost effective. It is not like, I'm pretty sure you guys have done this. Some of you all might have used this method. Very, very simple. Reagents are also not very expensive, so you can use that. Then for minimum inhibitory concentration, you can use the broad dilution method. You can have the tube macro dilution method. You can also have the micro titer micro dilution method as well. Um, and then you have got this one, which is the agar dilution method. So in agar dilution method, what is done is basically you have got uh, you have got different sorts of plates, right? You have got different sorts of plates or different concentration of plates, and generally the concentration is like, you know, 0.1, 0.2, 0.4, 0.8, doubling. Okay, it is a doubling concentration generally. And what happens is, again, you are doing the zero point 0.5 McFarland standard based on which bacterium you are using, or how many strains you're using. And you can see on the picture on your left hand side first. Okay, you have got that steel kind of thing with some wells, right? You're able to see that, right? Vaguely, you've got some wells over there. So in those wells, that concentration of individual strain is put. And what happens is that you have got the steers replicator or the inoculator, which is the second picture or the central picture that is there. So what will happen is that it goes, there is a button on the side. So you press that button, so it goes, it sits in that, in those wells. We call them wells, in those steel wells. It will sit in those wells and it will lift itself up, and then you put a plate in front of it. So then in the third picture you see. So that is the plate that has got the different colonies. So it will go and it will imprint 1 microliter on that particular plate. And so on you will see at, different concentrations. So you can determine the MIC, where the inhibition is occurring, that concentration where you will see no growth of strain number one or whatever two or whichever, that will be the minimum inhibitory concentration. So this is a very gold standard method, agar dilution. I'm not sure in India if you guys do or you don't, but whenever I did my PhD or if I do anything and if I have to be very sure, certain and everything, then I usually do the agar dilution method in order to be very certain, very confident that okay, it's resistant, sensitive. And you can also have strains over there that are sensitive as well with, already the MIC for those. So then you interpret that. First of all, you will come to know whether you have made the plates correctly or not. That is the first thing. And then the second thing is you are going to interpret your results from that as well. So that's why controls are also important. It is not like for fun we use control. No, you need to understand why you are using. Then it is easy. So you have got another, we like to make life easy, more easy every time. So this is e-test. So e-test is also used very frequently. E-test, still I think it is expensive. It is not very cheap method. I have used e-test myself. But you can see two examples over there. You have got the strip, okay? So same thing you are doing. You are taking the plate and you are swabbing the plate with the bacterium or with the fungus, for example, at whatever McFarlane standard, 0.5 McFarlane standard. And once you do that, you have the strip from the lowest to the highest concentration kind of. And I told you earlier, it is a doubling concentration gradient that is there. You place it on the agar surface and you incubate overnight at 37 or at whatever the optimum temperature is, with whatever condition, aerobic, anaerobic, microaerobic, depending on the pathogen. And you will be able to see this kind of a epsilon shaped zone. So that junction where you will see the growth is stopping, that is the minimum inhibitory concentration. So that concentration, if you do the yeast test for one bacterium, and if you do the minimum inhibitory concentration using agar dilution method, you will see that it will match the same concentration. There is no difference in that. It will be maybe one or two levels up or down, but it will ideally be matching on that side. So how do AMR genes mobilize? We know what is mobilization, so movement. So first is vertical transmission. Everyone knows what is vertical transmission, right? What is vertical transmission? Anyone else? What is vertical transmission? First, something actually say that I'm an ADHD. In order to lower says the vertical transmission on the dynamic. So there is vertical transmission, vertical. Okay, as the name is saying. Horizontal, horizontal. It is in the horizontal direction. So in horizontal transmission, remember three methods are there. One is transduction. Okay. Transduction happens when you are having the bacteriophage. Bacteriophage is a virus that infects bacterium. Bacterio stands for bacterium and phagos means to eat. Okay, so that is a bacteriophage. So you can see that again in the picture. You can see the phage, which is blue in color with the red head. And you can see that the phage will come, sit on the bacterium, it will release the genetic material, and the genetic material will go inside the bacterium and it will hijack the cellular machinery of the bacterium and it will create more and more bacteriophages. So that is transduction. Okay, remember that. There is, I can't give you easy way to remember, unfortunately. So there is transduction, transformation. Okay, transformation. You have got bacteria here on the table. You have got bacteria over there on the seat. There are dead. There are other bacteria that are alive. Okay, so when dead bacteria are there, the DNA is there around it. So if it finds the DNA beneficial, it will take that DNA. That is transformation. The bacteria is getting transformed into something new. Okay, so there is uptake of DNA. Some bacteria are very, very excellent in transforming themselves. They love that they always like to change. When you go for party and everything, you will have like new dress and it's the same situation over here. They are having different sort of genetic makeup, changing constantly, rapidly. Streptococcus pneumoniae. If you want to learn of transformation, read a lot about Streptococcus pneumoniae. It is one of the classic organisms for transformation. So there is transformation, and then the third one is conjugation. Okay, 99% of the people on Earth only think horizontal transmission, meaning conjugation. That also is, I don't know, what block is there for students or anyone other people. But it is not only conjugation. Conjugation is part of horizontal transmission, but other ways are also there, transduction, transformation. Horizontal, you can see, nicely in the picture. When vertical is there, it is showing vertical. When it is showing horizontal, you can see horizontally it is transmitted. So conjugation is nothing but it is bacterial mating. When two bacteria, mate with each other or they have sex with each other, when they are, uh, transferring the material or the plasmid, for example, from one bacterial cell into another bacterial cell. So the one where the plasmid is there, that cell is called as a F plus cell because it is fertility plus. F is fertility factor. It is called as fertility plus. So that F plus cell when it is transferring from that one to another one, the F minus cell, it becomes F plus cell. That is how it is. And remember, conjugation is not like, oh, any bacteria will mate with each other. In bacteria also we have compatibility and incompatibility. Okay. So it's not like any bacteria will have, sex with any bacteria. No, it is not like that. There is very specific, rules that are there already. Rules in the sense of we have not established bacteria themselves have evolved in a way where they can, do this mating with each other, where they can transfer resistance genes. And you can see that certain genes are transmitted from positives, gram positives to gram negatives, but you will not see very commonly from gram negatives into gram positives, for example. But again, in between gram positives, you will see lots of again transfer that is happening with other resistance genes. So that plasmid incompatibility is another thing. The plasmid incompatibility is nothing but, it is not like they will just mate because they want to mate. No. So that is another thing to bear in mind. And if you want to read, you are free to read. I'm just giving you some extra things. If you are interested, you can read what is zinc compatibility and how does it affect, because that plays a very important role in evolution and other factors, which is another area of mine that I study. Um, and then obviously we have got the mobile genetic elements. We have got the plasmids that I have already highlighted, can pass through conjugation, often harbor AMR genes, can often harbor heavy metal resistance genes, especially people who are doing wastewater now, people already know lots of resistance genes, that are cadmium resistance, mercury resistance, and other things. So these can also be detected through molecular methods. Okay, so how do AMR genes mobilize? So I already gave you an example of vertical transmission, already highlighted, transduction, conjugation, and transformations. So I'm not going to go over all those things again. For mobile genetic elements, we have got the plasmids, I told you already. Plasmids can be circular, plasmids can also be linear. Okay, exceptionally, they can be linear as well. And plasmids are something that don't require cellular machinery and all. They are independent, they replicate independently. Um, and I told you previously as well, they can have a broad host range, which is basically they can, one type of plasmid can be found in E. coli, another can be found, similar can be found in Acinetobacter, and so on. And that also depends on incompatibility and compatibility as well. Then we have got transposons, right? People here might know what transposons are, but you don't know what transposons are. These are elements that have like insertion sequences or insertion elements linked to certain structure, and these are jumping genes. These insertion elements, insertion sequences, these are jumping genes that can move within and between DNA molecules. So you have got the bacterial DNA, and in the bacterial DNA, you have got, a transposon that is there in between. So transposon can be harboring a functional resistance gene in between it, for example. A transposon can be an insertion element can be present upstream of a antibiotic resistance gene. So there are different types of transposition. One-ended transposition is there. So many things. You might, if people here have used the Excel function, I'm sure you know what is the transpose function. All the Excel experts over here. Transpose function is from, you know, changing the alignment from one place to another. So if it is vertical, you make it horizontal. So that movement is there. So often carry AMR and other genes. So it is present on plasmids, also on chromosome. And some examples are the TN3, TN21 family. I have discovered some insertion elements as well in my career as well for Acinetobacter. And my whole PhD is on that transposons. And if you are again fascinated with transposons, then you should read the work of Barbara McClintock. She is another great woman. I will not tell you everything about her, but she is a great lady who overcame lots of barriers in her life and was awarded an unshared Nobel Prize. She is one of the few ladies to be awarded an unshared share of a Nobel Prize for her discovery on transposons, where she was highlighted. She was working on maize that time. So read about her work. It is very interesting. So insertion elements I already told you. So IS26, it can flank resistance genes, facilitate rearrangement. Then you have also got integrons. So integrons were discovered by Ruth Hall, if you don't know. Ruth Hall is again, she was a professor in University of Edinburgh. I'm not sure if she was my supervisor student or not, but she was there in Edinburgh University when she discovered these integrons. So integrons are these small gene cassettes that have AMR genes, and there is an int-I gene, which is the integrase gene that is there, and it can excise itself. That cassette can come out and it can go somewhere in other bacterial species, and it will have a resistance determinant across it. So they can be embedded on transmosons or plasmids as well, these integrons. So next one is mobile colistin resistance. So mobile colistin resistance, you might have heard about this, but this is known as MCR1 gene, for example, or MCR gene is a gene that confers colistin resistance. Mobilized by ISAPL1 via composite transposon TN6330, highly recombinogenic in natural population. So this means TN6330 can lose one or both of IS copies, resulting in loss of transposability and stabilization of the cargo gene. So below in the picture you can see the ISPL1 flanking the MCR1 gene. MCR1 gene is in red, and on the side you can see the insertion element that is there, right? So that is a composite transposon, and you will see in the next slide about you'll learn about that. So it can go and it can put itself in the chromosome, then it can excise one portion of it, and it can do all sorts of things. It can also be present on plasmids as well. So transposable genetic elements are important source of AMR genes found in nature. Uh, compound structures occur from random insertion or of two insertion element copies on either side of passenger or cargo genes. Sometimes a surrogate IS can be used instead of a direct copy. And observe this figure now nicely. You can see structural gene. Structural gene can be an antibiotic resistance gene, virulence gene, anything it can be, whatever is needed for the bacterium to move around. And on the side, you can say highlighted in yellow and in red. You can see those are the insertion elements. Okay, those are the insertion elements. And the red portion that is there, those are the inverted repeats. When I say inverted repeats, they are mirror images of each other. Okay. So the yellow one that is there, that is the genes needed for transposition. Okay, so this, if you see structure like this is called as a composite transposon, and that is what you saw in the previous slide as well. Okay, this is a simplified version. So next we talk about Acinetobacter species or A. baumannii. I already highlighted that it is a gram-negative aerobic rod-shaped bacterium, non-motile, non-sporulating, capsulated, pleomorphic, and it has got, I told you Acinetobacter is a bacterium that is not motile as such in the true sense. You know what is motility, right? Flagella is the one provides motility. It doesn't have flagella or anything, but it provides what is known as the twitching motility. There is gliding, sliding, twitching, so many different types of motilities are there, and different bacteria exhibit different types of motilities. So this one exhibits twitching motility. Polar fimbriae, okay. Habitat, I already told you that hospital-related strains are very epidemiologically distinct. It is a critical priority pathogen, and increase in carbapenem resistance is what is seen in this specific bacterium, and it leads to more than 250,000 deaths each year. So to understand beta-lactamases, we will understand. Just be quiet. For a long time you are talking, so I can't concentrate. So we have got the beta-lactamases, and we can see class A, B, C, and D, right? So these are the four main classes. In class A, you have the serine at the active site. At class B, we have got zinc at the active site. Class C and D also, you have got the serine amino acid at the active site. In class A, mostly there are ESBLs. ESBLs are extended spectrum beta lactamases. Okay, ESBL, extended spectrum beta lactamases. And examples of those are, CityXM is there, TEM is there, SHV is there, JES is there. I know the whole names of all these short forms, but I'm not going to tell you. It will cause you headache, really. But I know that because I told you, I have been studying genes like ABCD, like letters of the like the alphabets basically. So we have done lots of work on these genes over the so many years, 17, whatever, 18 years. So extended spectrum beta lactamases are the ones that are going to hydrolyze, all these cephalosporins, penicillin, those kind of antibiotics. In B class, you have got the metallobeta lactamase. These are carbaminomases. So they are going to hydrolyze carbapenem class of antibiotic, imipenem, meropenem. So NDM, IM, VIM, these are all metallobeta lactamases. In C, the ADC is there. That is Acinetobacter derived cephalosporinases. So again, they will hydrolyze cephalosporin class of drugs. Ceftazidime, ceftaroline, ceftobiprole, uh, cefalozine, cefuroxime, all these drugs. And in class D, you have got oxacillinases, you have the oxa 51, and then other classes are there. So 23 that is there, oxa 23, 24/40, 58, and then oxa 134. Those are all carbapenemases. Oxa 51 is not a carbapenemase. Okay. Oxa 51 is the intrinsic gene of Acinetobacter baumannii only. Okay. Acinetobacter baumannii is identified based on that gene. If it is positive for oxa 51, it is a baumannii. 99% of the species will have that gene. And my supervisor is the discoverer of that class. Not only that, he also discovered oxa 23, which is another major carbapenemase now. In the days when he used to tell everyone that, oh, resistance is increasing, funding is not there for these kind of things. And now suddenly there is a lot of funding for AMR because AMR is a very important area nowadays. So, yeah, I remember him telling how they were competing with a French group during that time for this discovery of the class. Because that time, no whole genome sequencing, nothing was there. Only molecular methods were used for identification. So one of his students, she identified that Susan Brown is the name of that student. So you can read that review if you're interested. There's review on the right hand side again. It's a very nice review by Ben Evans. He's a colleague of mine. Um, so this is highlighting international clones and OXA 51 like genes associated with them. So just to clarify, international clones are also classified as European clones, worldwide clones, global clones. It is the same thing. And clonal complex, if I say, then it is like an umbrella. This is a clonal complex on top. And under the clonal complex, you have got different strains that are there. Okay, different strains are there under that. They are grouped together. So they are epidemiologically linked in that. Like in our family, we have the family tree, no? In the family tree, we are linked. So we are closely linked, like brothers who are there, they will be having close relation with each other. Sisters that are there, they will be having the close relation with each other. So in the same way, in epidemiology, this kind of linkage is there. So you can say up to 11 lineages exist, or 11 lineages meaning the clones of that particular lineage. And you can see the distinct OXA 51 variants that are linked in each of the lineages. So what you need to remember is that 1, 2, and 3, these are the main lineages that were first discovered, So International Clone 1 has the OXA 69 gene, International Clone 2 has the 66 gene, and 3 has 71 genes. So previously these used to be called as European clones because we did a lot of work in European countries and this was highlighted. So you can see the phylogenetic tree that was constructed by one of my students some time back. which is nicely separating out, IC3, IC1, and IC2. Okay, so there are another sub-variants over there. So that Oxo 69 that is there, Oxo 66 that is there, Oxo 71 that is there, those are Oxa 51 like genes. Okay, because Oxa 51 like meaning it is similar. That is the broad term that is used. And under that, each variant that is classified is given that number 69, whatever the number is 66, 71, 94, and so on. So you can see that in the international clone 2, that is the most widespread clone of Acidobacter in the world. And you can see that there are three alleles that have synonymous mutations. Okay, synonymous mutation meaning that the amino acid is not altered. When you take the DNA and convert it into RNA and then into protein, you do the conversion. The amino acid doesn't change at any position. So there are three alleles. So these alleles are based, alleles are nothing but they are variation of a gene. So these alleles are based on a database that is there. And some of the strains, they have ISABA1, which is, I'm sure there is any insertion element linked with OXA66 gene. So OXA66 is the core gene of clone 2. And then so on. You can also see others that are having like transposon structures, TN2009, 6, and 2008, and so on. And you can read more about this paper like Negro and Hall if you're interested. But OXA23 gene that is there, that is a very important carbapenem resistance associated gene that is linked with transposons. So in the data set that I had analyzed, or the student analyzed earlier, she saw that a lot of isolates had the TAM gene, which is ESBL class A. Okay. And the fluoroquinolone resistance was very, very high. You can see that 97 and 94%. And the MLST that is there, so that is the scheme that is used for identification of strain. Like we have got our, everyone has got a passport, right? And everyone has a name and a surname. So in the same way, if you say it is ST2, it is ST4, it is isolated, it is characterized at the genetic level. At the genetic level, exactly it is 1, 2, 3, and then you can link it. You can put it in this particular clonal complex. You can put it in this particular lineage, in this ancestry. So you can understand evolution and you can understand what happened in the past, whatever years, 100 years, 200 years. So core genome MLST is also there, so core genome MLST is looking at the core genes, entire core genes of the bacterium, and that can also be used. That gives a good indication of, which strains are clustering more together. It's like you are doing one test in your family between you and your sister or your brother or your mother and father, and you are understanding, okay, you are related with them, right? So that one test that is done, that is the MLST test. But if you are doing a more sophisticated test where you are looking at, I don't know, 200 genes from you and your mother and all, and trying to understand, or you have five sisters that you want to see, okay, with which one I'm more closely related or have close similarity, then that is core genome MLST. Okay, I understood. Don't be confused. I will give you all sorts of examples. So that's important because it tells you about the evolutionary history and characteristics. Okay, what has happened, how, and sometimes it also happens in our family. You will resemble. For example, for me, they say, or for my one of my cousins, they say like he resembles very much like my mother, although he is my cousin, for example. So that is all because of the genetic makeup, right? That is what is taking place over here. So in the healthcare sector, obviously we need, antibiotic stewardship programs. We need like infection prevention and control. surveillance and reporting and education and training. All these things are needed, if you want to improve and if you want to get on top of AMR. In the agricultural side, obviously, I need not tell you, you already know, limiting the use of antibiotics in the sector, maybe banning them or restricting them, and using under veterinary supervision, for example, using improving animal husbandry practices, enhancing biosecurity, hygiene to reduce disease incidence and transmission, and tracking resistance trends as well in zoonotic pathogen, trying to understand, what kind of resistances are seen. Because in animals also, it's not like those pathogens are not resistant. And the antibiotics sometimes that are given over there, in animals, they are slightly different, but similar forms of antibiotics we also take, no? So that cross resistance is evolving in that way. We need to prevent that. Policy and governance is important. So regulation of antimicrobial sales, you know, again, like counterfeit, for example, counterfeit antibiotics are there sometimes. You need to know that, okay, which ones are correct ones. And the national action plans are needed. Many countries have national action plans, and then funding research in that direction to focus on these public health initiatives. And lastly, sharing the data, especially in the neighboring, at least in the neighboring regions, I think it's very, very important that, the countries that are there, because what is happening, we are surrounded by other countries, no? So whenever we are there in the UK, we share the intelligence with the other countries. I have been to the European Center for Disease Control and Prevention, and I have seen how this happens as well. I had the good fortune of going and sitting in that big panel where they are monitoring threats of diseases. So in that way, that coordination must be there between different countries and understanding as well. That, okay, what we need to do, how we need to build the capacity to improve um, those things. So research and innovation, obviously, I don't need to tell you that we are in the dire need of R&D, novel antimicrobial phage therapy and vaccines, and also rapid diagnosis. So the rapid diagnosis, I know India is doing extremely good in the rapid diagnostic side of things. There are lots of nice things that are going on. So we need to keep on continuing to innovate in that area, and also do things in a way that are applicable not just to the people who are having, you know a lot of wealth as such, but also the people who are in the who are not having that kind of affordability, things like that. So in order to meet the demands of everyone, because health is there for everyone, no, it is not just a right of, the ones who have the money. No, it is a need of everyone. So we need to innovate in that way, that the methods are cost effective and they can impact those lives of people who are, in need. So this is the another example. Phage therapy, you might have heard already, but it says that research paper says la action bactericide de la Yamuna et du Gange sur le vibrio du choléra. Maybe you don't understand what that is, but what it is saying is that the action of the bactericide that is there from the water of Jamuna and Ganga on Vibrio cholerae. Okay, so bacteriophage was discovered in India. Did you know that? How many people know that bacteriophage was discovered in India? Okay, few people know. But this man, Ernst um, Hanbury Hankin, is the one who actually discovered that. And then the other people are there, like Felix Darrell and Edward Todd, who highlighted. And in the picture, you see Stephanie and her husband. So, husband was infected with Acinetobacter, basically, severe form of Acinetobacter, and she was the one who got the phage for him to save his life, and it is saved his life. So, this is another true story. You can read about her work also, it is very interesting. She has got many books as well. So new antibiotics are needed without, I don't need to tell that to you, but this is another method Kim Lewis was the one who discovered this method or this technique, and you can read this paper where they are kind of making cells to grow um, new type of bacteria that were previously unculturable. And he highlighted an antibiotic called as textiobactin as a result of that, which was able to treat staphylococcus. I'm not sure the progress of it at the moment, but I do know that when it was discovered, it was in the news that textiobactin targets gram positives. And then obviously public health awareness campaigns, education campaigns. I work with researchers who develop comics. Like you can see, like my son reads these kind of comics on plasmids and other things. He even knows. I will make you hear his bacterial names. He knows what is Staphylococcus aureus. Is 3 years old son, huh? 3 years. So that is there. So education and then behavioral change, obviously. I do a lot of work in Nigeria and other African countries. Also do like translation, you can see over there for like Microbiology Society. I have done translation in different languages as well. So behavioral change, I think everyone, I need not tell you, you need to know that you need to do a behavioral change and to encourage everyone to do that, discourage self-medication and sharing antibiotics. And then lastly, ways to reduce AMR is reduce the unnecessary usage, good antimicrobial stewardship, um, you know, use chemicals that can displace um, antimicrobial resistance, plasmids, improve diagnosis using low-cost methods, vaccines, bacteriophage therapies, antimicrobial peptides, and then newer drugs can also be looked into. And obviously, hand hygiene and all these things by far is still very, very effective. We learned that from the work of Semmelweis and Lister. So that's it, I think, uh, from my side. And we will do a quick quiz now. All right. So take out your phone now, quickly. Enjoy.